From counting birds at Christmas to cataloging bacteria in the human microbiome, citizen science is a template of scientific participation that can be co-opted by teachers to engage large numbers of students in research training. Assisting with laboratory classes is one of the best opportunities for early career scientists to start their teaching. But designing these classes requires a different skill set altogether. How can you get ahead of the game and leverage citizen science to synergize your research with your teaching? If it's your first time here, my name is Jack Wang, a microbiologist, science educator, and the 2020 Australian University Teacher of the Year. On this channel, we talk about science education, laboratory skills, and professional learning for teachers, and all of these themes will converge over the next few videos in ideas for lab classes that promote inquiry-based learning through citizen science. If you're an early career scientist designing a lab module for the very first time, I wouldn't recommend immediately running a crowdsourced research project targeted for the general public. You can, however, design course-based undergraduate research experiences, known as cures, to enable large numbers of students to participate in your research project as part of their normal coursework. This series of videos will walk through lab techniques and research questions adaptable for semester-long undergraduate projects and demonstrate how you can adapt cures into citizen science. Today, we will start with recombinant DNA technology or molecular cloning, which is a common point of entry into inquiry-based lab classes. Students design PCR primers targeting a gene of interest, prepare the PCR reactions, set up restriction digest and ligations, and verify the cloning of these plasmids through gel electrophoresis and DNA sequence analysis. The sequential nature of these experiments highlights the importance of controls at each step, providing students with a system where troubleshooting and optimization are vital for success. If you don't have time to watch this video in full, the TLDW version is this. In molecular cloning, if one step fails, everything fails. It is very tempting to have students follow cookbook recipes for each part of the process, but it is more valuable for their learning if you give students more autonomy in experimental design. You can soften the learning curve by limiting the parameters that students can change, designing controls, optimizing concentrations for different reagents. All of this provides a scaffolded safety net for students to make inquiry-driven decisions. Lab class design is a big emphasis in science education literature, and you can look through JIMB, FAMBED, CB Life Sciences to see how others have incorporated recombinant DNA work into research lab teaching. Useful keywords include undergraduate research, cures, or citizen science. I've included a link to a review I wrote on undergraduate research in the description below. That's it in a nutshell, but if you're still here, let's dive a little deeper. For each step in the molecular cloning workflow, let's go through how to introduce inquiry-based elements to challenge student thinking. Starting with PCR primer design, you can give students a pool of different genes to choose from and ask them to design PCR primers to amplify them. You provide basic design principles about primer length, overall G+C percentage, and melting temperature ranges, and students have to work it out. If you have the time and resources to order and test these student primers, that's great, but not very scalable for large classes. A workaround involves 3 different sets of primers that you've already designed for every gene, and asking students to use design principles to predict which primers will work best. Another exercise would involve a set of primers completely designed by an online tool without any manual tinkering. Can students beat the algorithm? Will their primers be better than the ones automatically generated by a computer? The next step is to actually run the PCR reaction and analyze the PCR products using gel electrophoresis. But I don't think this is a great point to introduce too many variables. Of course, the annealing temperature will change if the primer composition changes, but each PCR reaction could take a couple of hours to run. Not the best use of your time waiting around. I will keep the PCR conditions as consistent as possible so you can fit as many reactions into the same PCR machine as possible. Students should make their own PCR master mix and do the calculations for each reaction, design positive and negative controls for the PCR reactions, and predict the size of DNA bands to be amplified. This can then be compared to the gel electrophoresis results, which should be a standard operating protocol throughout these lab modules. Next up is the restriction digest. Most labs will provide students with one consistent cloning strategy to use just because there are too many ways this can go wrong. A slight compromise is giving students a choice of 3 different cloning strategies, sets of restriction enzymes, or plasmids with different tags, GFP, his tags, GST tags, and asking students to justify which will work best. They need to look at the multiple cloning sites of the plasmids, the sequence of their genes of interest, as well as any PCR primers you provided or they designed to see which will work best. When setting up the actual restriction digest, again, it comes down to students designing appropriate controls. Most likely a negative control in this case, and analyzing the outcomes using gel electrophoresis. After verifying the restriction digest and cleaning up the reaction, ligating the gene of interest and plasmid together is next. Ligation reactions are notoriously finicky, so ask students to take ownership over how many replicates to set up, the different ratios of plasmid to gene insert for each ligation, and again the appropriate experimental controls. All this then goes through DNA transformation into competent bacterial cells before subculturing any transformed colonies and screening using colony PCR. If the genes of interest are of genuine value to a research project, then there is vested interest to validate any positive clones. This makes the whole project more authentic, and one positive clone per gene can then be sequenced for them to check the accuracy base by base. We have separate videos on DNA transformation, colony PCR, and DNA sequencing on the channel, all linked below. If you add up all these steps, designing PCR primers, PCR reactions, restriction digests, ligations, transformations, this will take a minimum of 5 3-hour sessions for students to do everything. Maybe even more if you provide them with more responsibility in experimental design. This isn't glamorous work, but as molecular biologists, we know it is foundational to our field. How can you then get buy-in from students and leverage the 3 components of citizen science, authenticity, scalability, and communicability, to make the whole process more engaging? By focusing on the foundational lab techniques in molecular cloning, you've already made the whole module more authentic. Modest cost of reagents and wide availability of PCR machines and agarose gels improves the scalability of molecular cloning lab classes. What about communicability? Broader interest has to come from the genes students are working on and their connection to a disease or biological process. There have been many case studies in science education literature. Within my area of microbiology, cloning antibiotic resistance genes is very common. And a while back, I published a lab module looking at novel vaccine targets against bacterial urinary tract infections, again in an undergraduate research context. My go-tos again are JIMBY, BAMBED, and CBE Life Sciences. You can quickly look through these journals' back catalog to find examples of citizen science. There it is, a whirlwind tour through molecular cloning and recombinant DNA work in the context of cures and citizen science. Next time we'll focus on a different set of lab techniques that can also be adapted for inquiry-based learning and undergraduate research. This is the Biolab Collective. I'm Jack Wayne, and I'll see you in the next video.